Awesome, thanks for coming. For those of you who don't know me, my name is Jess. I am a neuroscience junior at Trinity, and I spent the fall semester of my junior year abroad in Buenos Aires, Argentina, and my internship was in the School of Medicine at the university. So the title of the talk is Biology in Buenos Aires, the Importance of the United States in Argentine Science. So this is just a quick overview of the talk. The first half will be about the actual research project that I was working on while I was there, and then the second half will be kind of an analysis of my observations while I was there, along with some background information with a focus on the importance of the United States in Argentine science. Okay, so to begin, my research project was the role of the RAS-MAPK pathway in stress tolerance. And this RAS-MAPK pathway has multiple roles. It's associated with certain positive and negative effects. So some of the positive effects are proliferation, long-term memory formation, and longevity and stress resistance. Some of the negative things that it is associated with are cancer, mental retardation, and cell death. So for this talk, and for the actual research project that we did while I was abroad, we're focusing on the positive effects of the pathway, specifically on the longevity and the stress resistance that it's associated with. Okay, so here's a picture of this RAS-MAPK signaling pathway. And for those of you who don't study biology, basically what it is a chain of proteins. So here at the top we have this receptor tyrosine kinase, which it's a receptor for a certain outside growth factor in this case. Then what happens is when this receptor is activated, we have a chain of effects, and the original signal is amplified and passed all the way down through the pathway to end up in the nucleus, and then the result of this pathway signaling would be the turning on or the turning off of gene transcription. So really quickly, these proteins right here are kind of docking proteins. They're not super important to the pathway, but what we actually studied was, we did study one of these docking proteins. This is the SHP2, and I'll talk about that in a second. What we know about SHP2 is that it's necessary for the function of the pathway, and we know that it works upstream of this RAS protein. So then once these docking proteins are activated by the receptor, this RAS protein is activated. This is a GTPase, so it binds and hydrolyzes GTP. And when it does that, it's activated. And when RAS is activated, it'll activate the rest of the pathway. So I won't talk about RAF or MEK at all, but then the next part of the pathway that's really important is this ERK, which is, I'm going to call it MAPK, it's the same thing. So MAPK is a mitogen activated protein kinase, and that would be the final step in the pathway before your gene transcription. Alright, so the foundation behind the whole experiment was this link between stress and aging. The idea is that manipulations that induce anti-stress pathways will then extend maximal lifespan. So the idea is if you're manipulating something and getting an anti-stress response, then you would also be improving longevity. So for our specific experiment, our immediate focus was cellular stress via heat shock. I'll talk about what we actually did in a second. And then the long-term goal, which is linking the stress and the aging, would be a development of therapies for aging. Okay, so we addressed two key questions in our experiments. And the first was, what is the RAS-MAPK pathway? What is its role in stress tolerance? And the second would be, out of the pathway that I just showed you with all of those different proteins, which specific proteins, or the genes that are making those proteins, which of those are actually providing the resistance to the stress, if in fact this does play an important role in stress tolerance. Okay, so to address these questions, we use the following experimental protocol. So first of all, we had these groups of flies, and they all had specific mutations. So we took tubes of 30 flies, and the first tube would have one mutation, and then the second tube would have a separate mutation, third, etc. So then you took all the tubes, and we placed them in a heat shock. We gave them a heat shock in a thermal bath. So the temperature was 39 degrees Celsius, which was pretty high. It's about 101 Fahrenheit. And we left the flies in there for 20 minutes. So then what happens is when you take all the tubes out of the thermal bath, they look like they're dead because the shock that we gave them was so intense that when they come out, it's like they're kind of in a sleep mode. They're not walking, they're not doing anything. It looks like they're dead. So then what happens over time is if they were able to resist that initial heat shock that we gave them, they will eventually recuperate in the tubes over time, and they'll end up walking around again. But if the heat shock was too intense and they don't have a strong stress resistance, they're just going to never wake up and they'll just be dead flies in the tube. So the way that we quantified this was through 5 different time points after the heat shock. So the first 4 were prior to the gradual recuperation, 0, 30, 60, 90 minutes. And the total recuperation we put at 24 hours. So that was, by that point at 24 hours, if they hadn't woken up, we pronounced them dead. So that was our protocol. All right, so back to the picture of the pathway. The first, we did a bunch of mutations, but I'm just going to focus on the 2 most important ones. The first is with this docking protein, the SHP2, and I'll show you the results in a second. And then the second mutation is of this MAPK protein at the bottom of the pathway. All right, so here are the results after the manipulations of the SHP2 protein, which was at the top, the docking protein. So just to orient yourselves, the bottom axis, the x-axis, was the 5 time points where we counted the flies that had fully recuperated, meaning they could walk on the walls. And the y-axis is the proportion of flies recovered. So if you're at 1, that's at 100%, meaning that the stress tolerance was incredibly high because all of these flies that we gave the heat shock to ended up recovering. So what we can see here is, first of all, I just want to point out that the control that we used, when we used the control, meaning these were the flies that had no mutation at all. This was a regular pathway. You can see there's no blue at all throughout any of these time points, so all of these flies remained asleep. They all died. Okay, so that's a regular fly under the circumstances that we gave them in the heat shock. And after the 20 minutes and the 39 degrees Celsius, they never woke up again. Okay, so it's pretty severe. So what's happening here is you can see that after we manipulate, these are 3 manipulations, I'll talk about them in a second. After we manipulate the SHP2 gene, there's a very high recuperation among all of these flies. So this red one, it's not super important, but it was just disabling the phosphatase activity on the allele. But the green bars are showing a loss of function. So that means that we downregulated the activity of that gene. And even as we downregulated it, you can see that the recuperation was very, very high, higher than 90%. I think that last one got up to 100%. So all of them are living, the stress tolerance is really high. The purple bars are the gain of function allele. So now we're